some of the first things to ever evolve on the planet. We're going to talk about their shapes, their arrangements, how you identify them, where they like to live. Okay. The archaea were probably the first organisms to appear on the planet, and that's because in the Earth's history, early on, the environments were relatively extreme. And archaea are extremophiles. They like to live in environments with higher low temperatures, higher low pHs, higher low pressures, higher low salt. Okay. And they're just fine with that. Okay. The bacteria likely came along afterwards. Okay. The bacteria are not as closely related to us as the archaea are. Okay. Which is kind of interesting. But bacteria have a series of shapes, okay? Some are round, like little BBs, and they're called cocci, okay? Some are rod-shaped, and they're called bacilli, and some are pleomorphic, meaning that they have a variety of shapes, okay? Let's look at some of these shapes. Let me set up the sound first. Okay, cool.
Prokaryotic microorganisms exist in a variety of different cell shapes. Some are spherical, but seldom are seen as a single cell. A spherical cell is called a coccus. Pairs of spherical cells are called diplococcus, the plural being diplococci. Chains of cells are streptococci, and grape-like clusters of cells are staphylococci. Some genera, such as Streptococcus and Staphylococcus, derive their name from the most common pattern of arrangement of the cells. Many prokaryotes are rod-shaped in appearance. A rod-shaped cell is called a bacillus. The genus Escherichia consists of rod-shaped cells. The genera Bacillus and Clostridium consist of spore-forming rods. Spore formation in some bacteria causes swelling of the mother cell. There are also many other genera of rod-shaped prokaryotes. Bacilli occasionally occur as chains of cells called streptobacilli. Cells of the Corynebacterium group have a characteristic club-shaped appearance. Some bacteria have a curved rod or vibrioid appearance. An example is the genus Vibrio. Mycoplasmas are a group of bacteria that lack a cell wall. The cells of this group are pleomorphic, meaning that the cells take on a variety of different shapes. There are two groups of spiral-shaped bacteria. Spirella are motile by means of flagella, and spirochetes are motile by means of axial fibrils located beneath a sheath that covers the entire surface of the cell. Bacteria such as the Nocardiae form branched filamentous cells. These cells reproduce by fragmentation. Actinomycetes form branched filamentous cells. This group is characterized by formation of a variety of specialized dispersal cells that form on the aerial mycelia. In some species, these cells consist of strings of individual small cells, whereas in other species, the dispersal cells are found in sacs called sporangia.
So you can see some examples here, right? On the left are some gram-positive cocci appearing as grape-like clusters. It's one of the most common causes of staph infections. Remember that the gram stain is one of the main ways we identify bacteria according to the composition of their cell wall. And that's why these appear purple. Next is E. coli, okay, a typical gram-negative bacteria, all right? And then you can see on the right, coxobacillus, okay, pleomorphic, and they can take on a variety of shapes. Several species of vibrios are human pathogens associated with gastroenteritis, cholera, food poisoning, and septicemia. Vibrio fischeri and Vibrio parviae are symbiotes of marine organisms such as jellyfish and can produce light through bioluminescence. The spirillum can cause rat bite fever, an uncommon but worldwide condition caused by rodent bites. These bacteria are present in the oropharyngeal flora of approximately half the healthy wild and lab rats, as well as other rodents. Spirochetes can also cause leptospirosis, Lyme disease, and syphilis. So you can see some examples here, right? Vibrio, lower left, spirillum, okay, and spirochete, all spring-shaped, okay, and motile by means of either axial fibrils or periplasmic flagella, all right? Then they move in a corkscrew type motion and frequently can tag tissues that they invade with decoy antigens so that the immune system is unable to effectively track down the pathogen the way it normally would. And it can also have residual effects such as autoimmunity. In addition to shapes, arrangements of cells are important, right? They can be categorized according to their arrangement and their style of grouping after cell division. Now, you might ask, why are we going on about shape and arrangement so much? And the answer is, it helps us identify what you're working with so that, for instance, in a clinical environment, you might have clues as to what the organism is and how to treat it with a particular drug, okay? Diplococci typically appear in pairs in one plane only. Example, strep pneumoniae, streptococci, bead-like chain, strepto means chain, okay, like strep pyogenes. Tetrads are produced by division in two planes, so they look like a box, sort of, right? Like beads at the corner of a box. So they're arranged in squares of 4. Sarcinae are arranged in cubes of 8 as a result of division in 3 planes, okay? And staphylococci are clusters of the cocci, okay? Diplo is typically going to describe cells that occur in pairs, all right? Individual species of streptococci are ID'd primarily on the basis of their hemolytic properties on blood agar. Strep pyogenes is responsible for strep throat, many cases of meningitis and bacterial pneumonia, endocarditis, and acute skin infections, as well as necrotizing fasciitis. These are the flesh-eating bacteria, okay? Micrococcus luteus, which can be found on many areas, including human skin, water, dust, and soil, shows this growth pattern. It's considered to be harmless, but some cases of micrococcus infection have been reported in people with compromised immunity, such as those on chemo or radiation, or those with AIDS, okay? So you can see examples here: staph, strep, micrococcus, okay? Bacilli, okay? Diplobacilli in pairs, right? Streptobacilli in chains. Again, indicating how they divide: 1 plane, 2 planes, and so on, okay? Bacilli tend to divide in 1 plane, producing diplobacilli and streptobacillus, okay? So you can see examples here. These would be gram-positive on the left, and these here would be gram-negative, okay? Bacteria divide through a process called binary fission, which is similar to mitosis in eukaryotic cells. It results in 2 identical daughters. Sufficient metabolites need to be there in order for division to take place, and then a cascade of events has to occur to initiate DNA replication. Once that's started, DNA synthesis has to complete, and then when the metabolites are depleted, it stops, okay? Bacterial replication is coordinated, accomplished by binary fission. At the point at which metabolites are depleted or toxic byproducts build up, chemical alarms are produced, and that stops synthesis. Degradative processes and DNA synthesis continue until all initiated chromosomes are completed, okay? So let's take a look at binary fission.
Cell division requires highly coordinated growth and expansion of all the cell's parts. Most Most bacteria have a circular chromosome that begins to replicate at its origin. At the origin, the DNA double helix begins to unzip, forming two replication forks. At each replication fork, DNA is synthesized by DNA polymerase molecules, which replicate the original DNA molecule, forming two copies. Replication proceeds in both directions around the circle. In a rod-shaped cell, the cell envelope and cell wall must elongate as well to maintain progeny of even girth and length. Growing bacteria replicate their DNA continuously with no resting phase. Sometimes even before replication terminates, two new replication forks form at each new origin of replication. Replication of the DNA termination site triggers growth of the dividing partition of the envelope, called the septum. The septum grows inward from the sides of the cell, at last constricting and sealing off the two daughter cells in a process called septation. Envelope extension and septation require rapid biosynthesis of all envelope components, including membranes and cell wall. Cocci, such as Staphylococcus aureus, do not elongate their cell walls before septation. Spherical cells expand their walls during septation. Here the furrows appear in the cell envelope all around the cell equator as a new cell wall grows inward. Two new envelope partitions are complete. The two daughter cells peel apart. The facing halves of each cell contain entirely new cell wall.
Bacteria in a fixed amount of media, like liquid media, exhibit a pattern of growth, okay, which includes a lag phase, a log phase, a stationary, and a death phase. Let's take a look at how that operates.
So in this video, I'm going to explain the 4 phases of bacterial growth. And so you may notice when you're looking at this, there are 5 phases. So there's the lag, exponential, stationary, death, and then long-term stationary. Most people don't talk about the long-term stationary phase as one of the main phases. So in this video, we won't talk about that as a phase. And in most of the classes that you take, it's going to just be the 4 phases up to here. So 1, 2, 3, 4. And so now let's go and talk about what exactly happens during these phases. So the first phase is the lag phase. And as you can see, this little bacteria is asking, where am I? So during the lag phase, the bacteria adapt themselves to the growing conditions, and they're going to synthesize molecules such as DNA, RNA, enzymes, as well as other molecules so that the bacteria can divide and reproduce. So in this situation, when they have enough nutrients, the bacteria are going to start preparing for growth. And so this phase right here, the first phase, is called the lag phase. Another thing to keep in mind with all of these phases, and particularly the lag phase, is that depending on the bacteria, this phase can take a varying amount of time. So it can go on for several hours or many days. It all depends on the particular type of bacteria and how quick the bacteria can replicate. And so one more thing I want you to keep in mind is that during this phase, there is pretty much no bacterial growth. So now let's talk a little bit about the next phase, which is the exponential phase. However, sometimes people call it the log phase. And so as you can see during this phase, the bacteria are growing really rapidly. So the amount of bacteria is increasing very rapidly. And so during this phase, there's not much going on other than the fact that they're just growing extremely rapidly. So the bacteria are dividing every couple hours or a couple days. But point is, during this phase, they divide very rapidly. And then eventually they're going to get to the point where the amount of bacteria being produced is equal to the amount of bacteria dying. So this usually happens when they start to deplete some of the nutrients that the bacteria needs to live. So as you can see here, during the stationary phase, there's no bacterial growth. And so what this means is that the number of bacteria are relatively constant. So these bacteria have essentially reached what we call a carrying capacity, which is essentially the maximum amount of bacteria that the population can sustain. And this is due to a limited amount of nutrients. So if we think about it a little more in detail, there's essentially two things that can occur during this phase. So your nutrients are going to be consumed, and then there's going to be toxic waste products that are released. So as a result, you can't have a continuous growth, a continuous exponential growth forever. And so as time goes on, more and more of the nutrients are going to be used up, and then as a result, some of the bacteria are going to start to die because there aren't enough nutrients. And so it's important to keep in mind that during this death phase right here, more bacteria are dying than are being produced. As a result, the bacteria population is going to decrease in size. And then it's also important to keep in mind that during this phase, some bacteria can form spores. And so that's essentially just like preserving themselves to be able to grow again when the environment is better. And then during this last phase, you have the long-term stationary phase, which pretty much no one talks about, so we won't talk about it. And that sums it up for this video. So I hope the 4 important things you get out of this video are that there are 4 phases of bacterial growth: the lag phase, the log or exponential phase, the stationary phase, and the death phase. Thank you for watching this video. I hope you liked it and found it useful. If you did, please be sure to give it a like. Comment down below if you have any questions or concerns, and subscribe if you want to see more videos like this.
So when we measure growth, there's a variety of ways we can do this, right? We can do dry weight, which is an accurate estimation, but it's time consuming. The cells need to be washed and then dried. Or we can do turbidity, which is how cloudy the liquid is, okay? We measure that with a spectrophotometer using visible light, and we measure it in optical density units. It's going to be greater than the cell mass. The greater the cell mass, the less light it's going to pass through. One of the things that is a drawback here, though, is that it doesn't differentiate between living and dead cells, okay? They can both contribute to turbidity. Another way is a direct cell count by microscopically examining the chambers. You use a hemocytometer. You put a little bit of the specimen at a particular time point in there. You count a specified area, and then you extrapolate to the entire growth flask. Viable counts require that you sample the culture at different time points and then plate them on nutrient media and then look for the growth of colonies and then extrapolate back with any dilution factor in order to determine how many cells you have at a particular time, okay? An example of a hemocytometer is here, right? There's a little grid on these little silver parts, right? Light passes through the grid, and you put a drop on there, you press the cover down, and then you spread these cells out, and then you count, okay? Several different slide counting chambers are available, okay? Petroff-Hauser, Sedgwick-Rafter, and Neubauer, but the idea is the same, right? You have a certain area represented by the grid, you count the number of cells in the grid, and then you estimate, okay? Now, you might say, well, great. Okay, so we're counting these bacteria. Why? What's the point? Okay, bacteria that are in log phase are easier to test the action of drugs on, and they're more effective at producing a desired gene product. All right, so one of the things you're doing with a growth curve is trying to figure out the conditions necessary to maintain logarithmic growth, right? When that happens, that's when you want to sample at what we call mid-log, okay? You don't really want to do a lot of work at the stationary phase where the death rate is equal to the growth rate, right? And you definitely don't want to do much in the death phase unless you're trying to generate spores, okay? Through a process called sporulation. So that's why we do this, okay? a variety of ways to get it. Now, are there other ways to count? Yes. You can use a cell sorter, which basically passes your liquid media through a pipette, and then a laser can detect the cells and can even differentiate whether they're alive or dead. Okay, so it's pretty fancy. Okay, high end. And you can also use PCR, where you amplify the genetic material present in the culture to determine how many cells you've got. And by the production of a particular gene product that that binds to a dye, right? The glow given off by the sample tells you how many cells you've got, okay? Because it's measured DNA synthesis. What are some factors that influence bacterial growth? Nutrition, temperature, osmotic pressure, and atmospheric pressure, okay? pH is another one, all right? The type of microbial life present reflects the physical and chemical conditions in an environment because of evolution and natural selection. Some microbes tolerate extreme temperature, high osmotic pressure, and other extreme environmental conditions. These conditions support the growth of one organism and inhibit the growth of others. All right. So again, evolution and natural selection is going to select for those traits that allow optimal reproductive success in that environment. And that's exactly what's happened with these bacteria that like these varying growth conditions. Okay. Phototrophs acquire their energy from light. Okay. Chemotrophs get their energy from chemical compounds. Carbon is the backbone for all organic compounds, right? Autotrophs get carbon from CO2 in the atmosphere, and heterotrophs get it from other carbon-containing compounds. Now, an interesting point here is the reason that carbon is the basis for all life is that it can do something that none of the other elements in the periodic table can do very well, which is it can form rings and chains with itself. And that's why carbon is the ultimate backbone for building tools of life, okay? If we look at photoautotrophs, they use sunlight as their energy source and CO2 from the atmosphere as their carbon source. Examples, photosynthetic bacteria, algae, and green plants, right? Chemoautotrophs use chemical compounds as the source of energy and CO2 as well, okay? Photoheterotrophs use sunlight for energy and organic compounds as a carbon source. The chemoheterotrophs use organic compounds as both the source of energy and the carbon source. So we are chemoheterotrophs, right? Photoheterotrophs can't convert CO2 into energy, all right? Some chemoheterotrophs, like Pseudomonas, can use many different organic compounds as energy and carbon sources, and therefore can grow in a lot of different environments, like soap dispensers. This makes the organism a particular problem in hospitals, where Pseudomonas can also grow in respiratory equipment, right? So one example of a particular problem, okay? Nitrogen, right? Some bacteria can obtain nitrogen from inorganic compounds such as nitrites and nitrates. Sulfur and phosphorus can be met by organic compounds or inorganic salts of sulfates, sulfides, and thiosulfates. The prefix thio means sulfur. Minerals are required in trace amounts, right? Nitrogen is an essential element for all life because it's part of a lot of workhorse molecules in living things like DNA and RNA and protein, just to name a few, okay? Sulfur, if soluble, phosphate salts are not available in the environment. They can be a limiting factor for microbial growth. Sulfur has to be as sulfates or sulfites. Okay, it has to be usually in the water table in the soil. Minerals required in trace amounts by microbes include potassium, magnesium, calcium, iron, molybdenum, manganese, and zinc. Small organic compounds are growth factors that are either not synthesized by a bacteria or are made in insufficient amounts. These growth factors include vitamins, amino acids, purines, pyrimidines, and porphyrins. Okay. This is a chemical that promotes growth. Temperature, okay. Mesophiles, optimal growth in moderate temperatures, generally between 25 and 40 centigrade. Habitat can be the soil, the human body, or animals. Most bacteria fall into this classification, and most pathogens, which we're going to be concerned with, fall into this classification. Thermophiles are heat-loving organisms. They grow best at 45 centigrade or higher. They're associated with hot springs, deep-sea hydrothermal vents, tropical soil composts. They are generally not human pathogens. Psychrophiles are cold-loving. They can grow at 0 degrees or lower. Optimal growth around 15, and they're associated with snow and ice. So you find a lot of those in Arctic and Antarctic regions. Psychrotrophs grow slowly at 0 degrees, but can grow between 25 and 30. They're abundant in nature and can cause food spoilage and refrigerator temperatures, and can be human pathogens. Okay, most microbes can grow out in a wide range of temperatures, but optimal growth is within their narrow range. Minimal growth temperature is the lowest temperature that supports an organism, and the maximal growth temperature is the highest that can allow it to grow. So what you have basically is a peak, and optimal is the top of the peak. It's the range of temperatures that provides the ideal conditions for microbial growth. In the lab, mesophiles are generally grown at 37 centigrade, which is human body temperature. Some thermophiles can grow in boiling water up to 110 centigrade. Psychrophiles are found in streams that are fed by glaciers. Okay, psychrotrophs are not considered to be psychrophiles, since they can also grow at room temperature or even body temperature. Right? Osmotic pressure is another consideration. Microbes with cell walls can withstand osmotic pressure. That's basically their bulwark against lysis, so they're osmotolerant. Osmophiles are microbes that require high solute concentrations in order for them to grow. Halophiles are salt lovers. Examples include halobacterium. Microbes with cell walls can withstand osmotic pressure, but most microbes don't tolerate environments in which the concentration of solutes is higher outside the cell than inside. Okay, in a hypertonic environment, they generally shrivel up, and that's called crenation. The water will leave the cell and move into the surrounding solution. If enough water leaves the cell, the cytoplasm shrinks and pulls the cell membrane away from the wall and can potentially kill the cell. This separation of the membrane from the wall is called plasmolysis, okay? And the process that produces it is simply dehydration, okay? Halobacterium and halococcus are present in salt lakes and basins where sodium chloride levels are close to saturation. Other obligate halophiles, meaning they have to have high salt to grow, are found in oceans and show optimal growth in up to 3.5% sodium chloride. Facultative halophiles can tolerate salt and live in salt concentrations up to 10%. Examples are Staph aureus and Enterococcus faecalis. Okay? Staph aureus is a common cause of nosocomial infections in hospitals in the form of MRSA. Okay? Hydrostatic pressure, right, is the pressure that a fluid exerts on a space. A barotolerant microbe can survive an increased hydrostatic pressure. A barophile grows best under high hydrostatic pressure and are only found in the deep oceans. Okay? Hydrostatic pressure is applied to aquatic microbes because of the weight of water. Most organisms, unlike those that are barotolerant, are inhibited in growth by hydrostatic pressures between 200 and 600 atmospheres. Okay? Basically 200 times to 600 times greater than atmospheric pressure. Obligate aerobes grow only in the presence of oxygen. They require oxygen to live. They obtain their energy through aerobic respiration, which is glycolysis, the Krebs cycle, and the electron transport chain all working together. Obligate anaerobes grow only in the absence of oxygen and are inhibited or killed by the process of oxygen. And so many of these are fermenters. Okay. Aerotolerant anaerobes can grow in the presence of oxygen, but can't use oxygen for energy. And these are obligate fermenters. And facultative anaerobes can do either or. Okay, they can grow with or without oxygen. It's important to know the oxygen requirements of organisms when obtaining, transporting, and culturing them. Metabolic pathways or organisms need oxygen to generate ATP, but some pathways can generate energy without oxygen. And some organisms will be killed in the presence of oxygen because of what are called reactive oxygen species. Okay. Obligate anaerobes obtain enough energy through anaerobic respiration or fermentation that they can survive, although they don't grow rapidly. Facultative anaerobes use aerobic respiration in the presence of oxygen and anaerobic respiration or fermentation without it. Okay. Microaerophiles require a low concentration of oxygen, about 2 to 10% less than atmospheric. Capnic bacteria require more CO2 than present in the regular atmosphere. Okay. Microaerophiles get their energy through aerobic respiration, while capnic bacteria, called capnophiles, also get it through aerobic cellular respiration, but they need this extra CO2 to grow. All right, let's break here for 15. So pH is just a measurement of the acidity of a solution. Okay, it's the concentration of protons in that solution, and every unit on the pH scale varies by a factor of 10. Okay, a neutral solution is by definition pH 7. Okay, anything below 7 is acidic pH. Anything above 7 is basic pH. Right. Natural environments have a wide range of pHs. Okay, 0.5 in acidic soils to over 10 in some alkaline lakes. The optimal pH for a microbe depends on where it lives. Neutrophils range between 5 and 8, acidophiles best below 5.5, alkalinophiles above 8.5. Most organisms grow best around 7. However, some species prefer an acidic environment. Helicobacter grows in the epithelial lining of the stomach, despite the high acidity present in the lumen, which is around 2. Thermoacidophiles grow at a high temperature and a low pH. Several groups of archaea are in this category. Some bacteria, such as Vibrio cholerae, prefer an alkaline environment and grow best as 8.5 or higher. Fungi are more tolerant of acidic environments and grow best around 5 or 6. So you can see the pH scale here, right? And notice, right, the alkaline and the acidic range. Some solutions are shown with their relative pHs. With increasing hydrogen ion concentration, the solution becomes more acidic and the pH drops. When the hydrogen ion concentration decreases, the pH rises. Okay. Every 1 unit scale in the pH scale is a 10-fold change in concentration. So if we were to ask the question, how much more acidic is something pH 2 than pH 4? All right, it would be 100 times more acidic, because from 4 to 3 is 10 times more acidic, and from 3 to 2 is another 10 times more acidic. So you'd multiply those together. Right? All right, let's look at some classification. Okay, prokaryotes are split into bacteria and archaea. According to Berge's Manual of Systematic Bacteriology, more is known about bacteria than archaea, which are a group of bacteria that have different features and tend to live in extreme environments, right? Not easily accessible by us, okay, and not involved in everyday human life. Bacteria, however, are our constant companions because they live on us, in us, and around us all the time. Members of the archaea are a group of bacteria that have features differing from the other bacteria, with the exception of thermoacidophiles, which are found in hot acidic springs. The rest of the archaea have cell walls that don't have peptidoglycan. Archaea are most often found in unusual or extreme habitats, and based on their habitats, they're classed as follows. Methanogens generate methane. Thermophiles love heat, and halophiles love salt. Okay? A bacterial species is defined as a group of bacteria that exhibit a large number of similar characteristics. Phenotypic classifications of bacteria involve microscopic and macroscopic morphology and were the first used to identify bacteria. Measurement of the presence or absence of specific biochemical markers, such as fermentation or specific carbohydrates, or the presence of specific enzymes, can be used for identification as well. Now, what we're talking about here, again, is this idea of using the classic methods of identifying bacteria, which have largely been replaced by molecular methods, okay? Classic methods use phenotypic characteristics, right? Growth on certain types of media, the shape and the arrangement of the cells and how they stain and so on, right? Molecular uses just the genetic material, okay? So we just sequence the genetic material, we compare it to the information in the database, and we know what we have, all right? Remember that when we're talking about classification, right? This was something generated by Linnaeus, okay? And from order of broadest to least inclusive category, domain, kingdom, phylum, class, order, family, genus, species. Dumb King Philip came over for good sandwiches. That's the way to remember it, okay? Linnaeus was the father of taxonomy, okay? Spirochetes are a group of helical multibacteria that move with a twisting motion. They're found in aqueous environments or in association with people or animals. Pathogens include Treponema palladium for syphilis, Borrelia burgdorferi for Lyme disease, Leptospira interrogans for leptospirosis. Spirochetes have a length of between 5 and 500 microns. The axial fibrils are a type of endocellular flagella that are wound around the body. The silver staining of Leptospira shown in this figure shows the highly coiled body with the hooked ends. Now, one of the things about these guys is that they wiggle, okay? They worm their way through tissue, and as they do, they can leave decoy antigen on the tissue, and that distracts the immune system and can cause a delay in the immune system finding the pathogen, and can even lead to autoimmunity. Okay, a perfect example of this is Lyme disease, which is caused by a spirochete spread by a tick bite. Okay, the tick bites you and you get the bacteria, and then the spirochete wiggles through the tissue and tags it with decoy antigen. And the result of that, right, is that we end up distracting the immune system, destroying healthy tissue that otherwise wouldn't be a target, which is no good, right? We hate that. And we can eventually clear the bacteria with a combination of antibiotics and immune system action. But in the course of doing this, you can end up generating autoantibodies that attack your own tissues. And so this is why a lot of times people that end up with an initial infection of Lyme disease can develop autoimmune diseases later, such as rheumatoid arthritis. Okay, we'll get into that when we get into those diseases. Okay? Aerobic or microaerophilic helical vibrio gram-negatives are either slightly curved or have multiple helical turns. They're motile with help of flagella. Some cause disease in humans or animals. Okay, this group includes Campylobacter, Helicobacter, Fidelovibrio, and Vampirovibrio. Okay. Gram-negative aerobic rods and cocci are a diverse group, extensively investigated, widely distributed. Many are human and animal pathogens. So a lot of bad guys in here, right? Respiratory metabolism, so they use oxygen. Okay, they don't generally do fermentation. You'll find that most human pathogens don't do fermentation as an exclusive way of getting energy, because they wouldn't grow fast enough and they wouldn't have as extensive an effect. Okay, some do though. There are some fermenters that cause human disease. Rhizobia are soil bacteria and are nitrogen fixers. Okay, Bacteria in this category include Rhizobium, Legionella, Escherichia, Bordetella, Francisella. They can't fix nitrogen independently, require a plant host. So they associate with what are called the nodules of legumes, okay, beans and peas, and they play a major role in agriculture. On the left is a gram stain of gram-negative aerobic bacillus. Notice it's pink, right? And on the right is a gram stain of gram-negative aerobic coccus. Again, notice it is pink. Facultative anaerobic gram-negative rods, a large group of bacteria with simple nutritional requirements. You find them in soil, water, and the GI tract of animals and humans. One example is E. coli, which is the workhorse of molecular biology. Okay. Bacteria found in the intestinal tracts of humans and animals are referred to as enterics and include Escherichia, Salmonella, Shigella, Enterobacter, Proteus, and Providencia. The best known enteric bacteria is E. coli, named for where it lives, the colon, which is an extensively valuable tool in genetic engineering and molecular biology. Right? Below, you can see gram stains of E. coli and Proteus vulgaris. Okay, so notice both are gram negative. Okay? Anaerobic gram negative rods can be straight, curved, helical, modal, or non-modal. They're present in the GI tract of humans and animals. Leptotricha buccalis and Fusobacterium species are often found along the gum line and the tooth surface. Bacteroides are normally commensal, representing a substantial portion of mammalian GI flora and preventing potential pathogens from colonizing the gut. However, some are opportunistic pathogens. Remember that a lot of your native flora, you know, the good guests in the hotel body, are actually protecting you against pathogens by taking away binding sites and generating chemicals that impede the ability of the pathogens to get a foothold and colonize you. Okay, so they're really another line of protection. And when we wipe them out by using things like broad spectrum antibiotics, we leave ourselves open for infection. Okay. More anaerobic gram negative cocci, right? Often in pairs, found in the mouth, in the GI, and the vagina. Veillonella causes opportunistic infections, often associated with longstanding gingivitis in dental patients. Cases of osteomyelitis, bacterial endocarditis, and abscesses in internal organs have been reported as well. Rickettsias and chlamydias, okay? They're small gram-negative bacteria, some of the smallest. Most require a host to reproduce. They're generally rod-shaped, but may look coxoidal. They often cause disease. Rickettsia need a vertebrate and an arthropod host, okay? And chlamydias do not infect invertebrates. Rickettsia rickettsii is the agent of Rocky Mountain spotted fever, and Chlamydia trachomatis is for UTIs, okay? Urinary tract infections. It's a host-dependent organism, often causes diseases in humans and animals. Orientia tsutsugamushi is an agent of scrub typhus, a widely distributed endemic disease in the Asian Pacific regions that was formerly classified with the Rickettsia. It's a gram-negative obligate intracellular bacteria belonging to the family Rickettsiaceae. Okay, so you can see an example of Rickettsia here, okay? These are little tiny purple guys, right? This is a Jimenez stain of tissue culture cells infected with Rickettsia. Okay, mycoplasmas and ureaplasma. The smallest free living bacteria, no cell walls, pleomorphic, meaning a variety of shapes, resistant to penicillin, cephalosporin, vancomycin, and other antibiotics that battle cell wall synthesis. Why? They don't have cell walls. Okay. They have special growth requirements and can cause disease in humans, animals, and plants. They're unique among bacteria because they don't have cell walls and have variable shapes. Mycoplasmas have special growth requirements, and most pathogenic species are host specific. Gram-positive cocci include aerobic and anaerobic bacteria, range from harmless to virulent forms, including Micrococcus, staph, strep, Peptococcus. You can see staph epidermis below there. Micrococci are aerobic organisms not associated with disease. However, they can be opportunistic pathogens in hosts with compromised immunity. These organisms are found in soil, water, dust, dairy products, and are part of normal flora in the skin. Staph are gram-positive facultative anaerobes and can cause a wide variety of diseases in humans and other animals through the production of toxins or tissue invasion. Staph toxins are a common cause of food poisoning because of the heat resistance of the anaerotoxin and the ability of staph to grow in foods with relatively low water activity. Strep are microaerophilic or facultative anaerobic organisms that produce lactate. Although a number of strep species are pathogenic, many are non-pathogenic and are part of the human flora of the mouth, skin, intestine, upper respiratory tract, and can be opportunistic pathogens. Peptococcus and Peptostreptococcus are anaerobic gram-positive cocci and part of the normal flora of the mouth, skin, and GI and urinary tract. They can infect normal hosts with compromised immune system or damaged tissue. Under those conditions, the organism can become pathogenic and can cause brain, liver, breast, and lung abscesses due to septicemia. The figure shows a gram-positive bacteria, which is a normal resident of human skin, which can become an opportunistic pathogen, and that's staph epidermis. Okay. All right, gram-positive rods and cocci include Bacillus and Clostridium. They're important in medicine and food industry. The spores are resistant to heat and disinfectants. They're widespread in the soil. In your text, page 98 shows a selected medically important endospore-forming gram-positive rods and cocci. The picture shows gram-positive spore-forming Bacillus cereus. Okay. Now, the spore formers are special, right? Because the spore is a way to go silent until good environmental conditions reemerge. Okay, they have managed to germinate spores in Arctic ice cores that go back to the Jurassic period when dinosaurs ruled the earth. So that's a long time to sleep and then come back to life. Okay, and I think we commented on the fact that every pathogen that we're going to look at in this class has a stage where it can basically hide. Okay. Viruses, we're going to find out, can become latent in some cases. Bacteria can form endospores in some cases. Fungi can form spores. The protozoa and the helminths can form cysts. Okay. And all of these are ways to wait out bad environmental conditions until good ones show back up. And then they could go back into what's called vegetative growth.
Yeah, no depression. No depression? I don't think so.
So let's look at some of these, okay? Clostridium perfringens is widely distributed in the environment. It's found in decaying matter, marine sediment, intestinal tracts of humans and other animals, and can cause food poisoning. The bacilli are either obligate aerobes, meaning they have to have oxygen to live, or facultative anaerobes, meaning they can switch off. Clostridia are mainly obligate anaerobes, meaning oxygen is poison to them, and include free-living and pathogenic species. Clostridium perfringens is important in medicine and in the food industry. In general, cocci don't form endospores. However, there are rare examples of coccoid-shaped gram-positive bacteria that, under the right conditions, can form an endospore, right? A way to hide until conditions get better. Non-sporulating gram-positive rods can be obligate or facultative anaerobes, have complex nutritional requirements, include Carnobacterium, Lactobacillus, and Listeria. Lactobacillus convert lactose and other sugars to lactic acid. Most are harmless. Some species are used in the food industry, such as making yogurt, cheese, sauerkraut, and pickles. Listeria monocytogenes produces listeriosis, okay? Corynebacterium pleistocenium was discovered in the Arctic part of Alaska. It was found in the permafrost, and melting ice brought these extremophiles back to a growing vegetative state. The discovery of this organism is of interest to NASA as they investigate the permafrost of Mars for bacterial life. Outbreaks of listeriosis have been attributed to the consumption of hot dogs, deli meats, raw milk, cheeses, raw and cooked poultry, raw meats, ice cream, raw vegetables, smoked fish, and has a high mortality rate compared with other foodborne diseases. Okay. A little bit about the species that are used in industry, right? Yogurt, lactobacillus can turn milk into yogurt. You can make yogurt in your home if you want by boiling milk and then adding a little bit of yogurt to it once it's cooled down. That will convert to all yogurt, non-flavored yogurt, right? Cheese is produced by the bacterial action on curd from the whole milk, okay? Well, we say the cream from whole milk, right? If you've ever had cheddar cheese, that name comes from the caves of Cheddar over in England, where the curd from the whole milk is placed while the bacteria that live in the cave convert that curd into cheddar cheese. That's where that comes from, okay? Sauerkraut and pickles, again, a lot of this is the result of helpful bacterial action, okay? So keep in mind that generally speaking, microbes are our friends, right? It's only the bad actors that we're concerned with here. Irregular non-sporulating gram-positive rods are generally pleomorphic. They're mostly facultative anaerobes with some obligate aerobes. Medically important species include actinomyces, which are opportunistic pathogens, Corynebacterium diphtheria, which causes diphtheria, Propionibacteria acnes, which causes acne, and Actinomyces israelii, which causes periodontal disease and lung infections. Okay. Mycobacterium are gram-positive aerobic acid-fast rods. They tend to form filaments. They're widespread in water, soil, and food sources. Some are obligate intracellular parasites in animals and humans, meaning they have to live inside the host to survive. They're tough to grow in culture. Why? Because they require host tissue to do that. They can colonize in the host without symptoms. Infections are difficult to treat. They're not really gram-negative or gram-positive. They're acid-fast. And they're naturally resistant to many antibiotics, especially those that go after cell walls, right? Because they don't have them. Although mycobacteria have a tendency to be difficult to grow in culture, they're classed into two categories: fast and slow growing based on lab growth characteristics. Okay? Propionibacterium shermanii is used in the production of Swiss cheese. So here we go with another beneficial organism. So you can see here an acid-fast staining of Mycobacterium smegmatis, right? This is one of the organisms that tend to grow in the anal genital area of humans, okay? This is one of the reasons that a lot of men are circumcised in the US to avoid infection by this very bacteria. Nocardioforms are aerobic gram-positive, catalase-positive rods that form branching filaments. They're all over the environment and opportunistic. Infection is rare. They originate in the lungs and can spread to other organ systems in an opportunistic infection. Although nocardia infection is rare, it's generally a chronic bacterial infection coming from the lungs and spreading to other systems. These other systems are commonly affected and include the brain and skin. Kidneys, joints, heart, eyes, and bones can be involved. The symptoms of infection vary and depend on the organs. Okay. Streptomycetes are gram-positive. They live in the soil and decaying vegetation and are spore formers. They're generally not pathogenic and infrequently cause disease, but they are an important source of antibiotics. They very infrequently cause human disease, most often manifesting as a localized chronic infection of the skin and underlying soft tissue. Invasive streptomyces is extremely rare. Okay. And finally, methanogens, methane generators, right? These are archaea that grow in anaerobic environments, such as swamps, marshes, hydrothermal vents. They produce methane as a metabolic byproduct and are killed by oxygen. Although methanogens haven't been considered to be human pathogens, Several research groups are studying methannogens as a potential human pathogen, especially in connection with root canals. Okay. All right. So let's take a little break here, 15 minutes.